If you're a dog breeder, genetics is basically your entire job. Even without knowing anything about the molecular basis for genetics, one would know that in order to maximize the success of a litter of puppies, whether it be in the show ring, on the job, or in a family, you would want to make sure that the parents of that litter also have similar characteristics. So how are traits like coat color, athleticism, trainability, or even genetic disorders passed down? The answer is deoxyribonucleic acid, or as it's better known, DNA. DNA is a complex molecule that is made up of many nucleotides stacked on top of each other and arranged in a double helix. These nucleotides come in 4 flavors, if you will: adenine, thymine, guanine, and cytosine. Adenine bonds with thymine, and guanine bonds with cytosine, which forms the double helix shape. The sequences of A's, T's, G's, and C's in the DNA codes for proteins. This is really important. Proteins make up almost everything in our bodies and our dogs' bodies. Little protein-making machines in the cell called ribosomes use RNA, which is a special nucleic acid which functions as the middleman between DNA and proteins, by looking at 3 bases at a time to assemble a protein. Each chunk of 3 bases, called a codon, codes for one of 20 amino acids. Stacking amino acids on top of each other is how the cell builds proteins. This is why DNA is often called the blueprint for all living things. Everything about everything that's alive, from you to a plant to a bacterium to your dog, is governed by this molecule. This is why sequencing and studying DNA is so powerful. We can find what makes our dogs the way they are, and this is how traits are inherited. Every dog and every human has 2 sets of chromosomes. Chromosomes are really long strands of DNA. 1 chromosome is from the mother, and 1 chromosome is from the father. That means you and your dog both have 2 copies of every gene in your genome. The interactions of genes on homologous chromosomes is what makes traits dominant or recessive. For instance, let's say 2 fictional dogs have puppies. The dam has a disorder in which the gene that codes for pigments in her fur that should make the dog brown like the sire is mutated on both of her chromosomes. The puppies will get 1 copy of the mutated gene that made the mother white, i.e. no pigment, and 1 copy of the wild-type gene, which is the word geneticists use to describe the genotype carried by the majority of the population. What color will the puppies be? In this fictional and oversimplified example, let's assume 1 copy of the wild-type gene is enough for the puppies to make sufficient pigment to give their coat a brown color. That would make the gene that codes for brown fur in the father dominant to the mutated white gene in the mother. This is essentially what dominant and recessive mean on a genetic level. The essential question is 1 copy of a wild-type gene enough for a wild-type phenotype? A phenotype is the outward expression of said genes. If it is enough, that wild-type gene is said to be dominant. If it isn't enough, it is said to be recessive. Sometimes it's a little more complicated, and it can get you halfway there, which is said to be incomplete dominance. Things get slightly more complicated when we look at genes on sex chromosomes. What determines sex in both humans and our dogs is whether we have 2 X chromosomes, which makes you female, or 1 X chromosome and a Y chromosome, which makes you male. This means that if a gene lives on the X chromosome, males will only have 1 copy of it no matter what. Let's use the same example from before with the white mutant dam and the brown wild-type sire, except this time, let's assume that the gene lives on the X chromosome instead of on an autosome, which is any chromosome that is not a sex chromosome. Let's make a Punnett square, which is a special diagram geneticists like to use to determine possible offspring of a mating pair. Punnett squares work using the idea that you get 1 copy of a gene from each parent, who each have 2 copies themselves. Therefore, a Punnett square shows all the pairings of the alleles between the 2 parents. You can see that all the males in this litter will be white, and all the females will be brown. When the mom passes down a mutant chromosome, only the females with a 2nd X chromosome are able to make enough pigment to make them brown. Mutations can be harmless, like making a dog white. They can even be beneficial and make a dog smarter or faster or cuter. But many times they can be really bad. This is what makes a genetic disorder. If a mutation messes up a gene that a dog needs to live a healthy life. Using what we learned, let's run through some of the key ideas, terms, and implications of genetics relating to your dog. Autosomal refers to a gene that lives on any chromosome that is not a sex chromosome. This means that it doesn't affect males or females any differently. Sex-linked means the opposite. It means that it lives on one of the two sex chromosomes, most of the time on the X chromosome because it's bigger and has more genes, but that isn't always the case. This means that a sex-linked genetic disorder affects males and females differently. Homozygous means that both copies or alleles of a gene are the same. A dog can be homozygous dominant, meaning they have two copies of the dominant allele, or homozygous recessive, meaning they have two copies of the recessive allele. It may be important to note that this can only apply to genes that have two copies, so a male dog cannot be homozygous for any sex-linked gene because they only have one copy. Heterozygous is the opposite. It means that the two alleles of the gene in question are different. Dogs who are heterozygous with one copy of a dominant wild-type allele and one copy of a recessive allele that causes a genetic disorder are often called carriers. This is because despite the fact that the dog is not affected by the disease because it's recessive, it can still pass that disease to its offspring, if their mate also happens to be a carrier. Recessive disorders, because of the fact that carriers are phenotypically normal, tend to skip generations. This is how healthy parents can have affected offspring. A large percentage of genetic disorders in dogs are inherited in this way. The thing that's good about carriers is that if a carrier is mated to a non-carrier, none of their puppies will be affected. This is how autosomal recessive conditions can be slowly eliminated from the population without losing too much genetic diversity. This is the power of genetic testing and the use of these principles in dog breeding. We can work to eliminate genetic disease in breed populations while maintaining the other characteristics of the breed that people love. When scientists and dog breeders work together, we can help make each generation of dogs happier and healthier than the generation before.